Dear students, today we are going to study about cell culture and isolation of single cells. Means how the cells can be isolated, single individual cells can be isolated for performing the cell culture system. Now, the concept of cell culture it starts when an interest was arised that how the cells when they were isolated from the mother plant how they would behave in vitro. Now, we know that in the wild certain plants they have the capability that when there certain small pieces of those plants they get detached from the mother plant, they in complete plant would be arises. For example, in case of dandelions, when their roots they get isolated from that isolated roots, you will get the complete plant. Even in case of begonia, the complete plantlet would be obtained from the leaf tissue. Also, the requirements for both the cell division as well as for the differentiation they differ, but the basic medium that is required for the basic growth it is the same one. It requires it includes micronutrients, carbon sources, sucrose, certain plant growth hormones, they should be there in this basic medium. Now, sometimes certain undefined components they are also added, which includes like fruit juices, plant extracts, even yeast extracts sometimes are also added. For example, it has been found that if in the orchid culture system, if coconut water or coconut milk is added, that would be beneficial. But the main drawback of using these undefined components is that its composition it will vary, it will not remain the same, and the components which are present in that you will not known. So the main benefit of this culture system of doing the cell culture system is that you will be able to investigate as you are using a single cell. So you will be able to investigate कि आपको पूरा plant की कैसे development होती है from a single cell. You could get to know it about it. Also, you can do the plant propagation with this cell culture system. Even in some cases, if a foreign gene is introduced with the help of genetic engineering, so transgenic plants could be obtained, which will be beneficial even for industries, and even that could have certain important or beneficial agronomic traits also. With the help of this cell culture system, virus could be eliminated so that you would be able to use healthy plants. Even the preservation the of the germplasm could be done with this. Even the plant species which are threatened, you could preserve those species also. Now, for doing the cell culture system, how the that cells can be isolated. Now, it can be done either from the intact plant organ, or even you can use the suspension culture system. Even in some cases, it has been found that the cells it could be obtained from the cultured system. Now, for obtaining the plants from the from in order to obtain the single cells from the plant organ, you can use the leaf tissue, that would be more beneficial. Now, for the isolation, either it can be done by the mechanical means or by the enzymatic means. Mechanical means may you will simply go with the grinding. Now, it has been in the year 1966, Colin Back, he first of all did the mechanical isolation from the Macalia cordata, which is a type of poppy. From that, he isolated the leaves, isolated the cells. Now, also in the year 1965 and 68, Joshi and Ball they isolated the mesophyll cells of groundnut, and that too in the leucan medium. And later on, two scientists Granum and Calandevelu, he both of these did the they prepared a grinding solution and grinded that leaves into that medium, and were able to isolate the individual cells for the culture system. And that grinding medium it consists of sucrose, magnesium chloride, tris chloride, and buffer was maintained. For the enzymatic isolation, certain enzymes they are added, which helps in the separation of the cells. But the main problem is that you have to add the osmoprotectant uh, into the medium, so that enzymes का जो भी कोई negative effect है that could be overcome. And the main osmoprotectant which is used is the mannitol. From the culture system, you can also obtain the individual cells, and it is much easier and even convenient to isolate from the culture system. Reason being, जो culture system है उसके cells जो होते हैं वो ज़्यादा friable होते हैं, तो easily आप कोई भी एक्सप्लांट लेके उसका कैलस obtain कीजिए, and from that individual cells can be easily obtained. Now क्योंकि जब आप continuously agitation करेंगे in the ल्यूकन medium from callus you can get the individual cells. Now the advantages of this mechanical method over the enzymatic method is कि आप mechanical method में कोई भी enzyme use नहीं कर रहे हैं, but in the enzymatic method you are using the enzymes. So other requirement, other main beneficial is that कि in the mechanical method also protectant जो हैं वो use नहीं करने is required. But the main disadvantage is that in case of the mechanical method, it has been found कि जो viability है cells की that is very less. So main disadvantage is this one. And the advantage of using the single cell culture system is that uh, in the single culture system क्योंकि आप एक single cell से you are doing this work, so इंडिपेंडेंटली आप एनालाइज कर सकते हैं कि एक इंडिविजुअल सेल क्या फंक्शन परफॉर्म करेगा, इवन उसका सेलुलर और मॉलिक्यूलर करेक्टराइजेशन जो है एक इंडिविजुअल सेल की, यू कैन स्टडी अबाउट दैट. अगर आपको एक कमर्शली इंपॉर्टेंट कोई प्रोडक्ट चाहिए तो यू कैन ईजिली गेट दैट फ्रॉम कल्चरिंग ऑफ ए सिंगल सेल, क्योंकि वहाँ पे और कोई सेल्स जो होंगे वो ऐड नहीं होंगे, सारे के सारे जो क्लोन्स होंगे वो एक इंडिविजुअल सेल्स के ही होंगे. इवन आपका कोई आपको स्टडी करना है कि कोई भी इफ़ेक्ट क्या है वेरियस सब्सटेंसेस का ऑन द सेलुलर रिस्पॉन्स, यू कैन ऑल्सो get to know about this. That was all about how the culture of the single cell, for doing the culture single cell culturing, you can obtain the single cell. That's all. Thank you.